All right, our last macromolecule of focus, nucleic acids. So with nucleic acids, what we end up with is the composition is a sugar, a phosphate, and a nitrogenous base. So it's a base that contains nitrogen. We're going to draw some diagrams in just a second. I'm going to show you some nucleic acids. You should know that you do not have to write anything on the product side. It says C below, and I'll show you what I mean by that momentarily. But one last time, right? Whenever we make a polymer, so when we go this direction, we're going to make DNA and RNA from nucleic acids, right? Individuals. When we make a bond, we do dehydration synthesis. What type of bonding joins things together? It's called a hydrogen bond. It's in the center. And likewise, if we went the other way from DNA and RNA, we would break it apart and call it a hydrolysis. Okay, here is the monomers. Here's the left side, the reactants that go in. So you can see your arrow right here. So you're going to write this part in. I told you didn't have to write the part in over here. But nucleotide number 1 has a phosphate, it has a sugar, and it has a nitrogenous base, plus nucleotide number 2, which has a phosphate, a sugar, and a nitrogenous base. And then I have this, plus additional nucleotides. You can have hundreds, if not thousands of nucleotides joined all together to make a DNA or an RNA molecule. And so this is our monomers that go into the chemical reaction. So you're going to want to take a second and pause the film and write this in. It'll take you a moment. These are the polymers that come out. It says here, you don't have to draw this one. It's actually located at the very bottom of your pages, but I do want to go over it. So what the polymer ends up being is either a DNA molecule, which means deoxyribonucleic acid, or an RNA molecule, which is ribonucleic acid. And the big difference between these two things is with DNA, we have cytosine, guanine, adenine, and thymine. And cytosine and guanine are complementary. A's equal, and adenine and thymine are complementary. So our A's and our T's will be equal. Our C's and our G's are equal. RNA is a little different. One of them is missing, you see. So we have cytosine still, we have guanine still, we have adenine still, but instead of thymine, with RNA, we get uracil. And so this, again, is at the bottom of your pages. You don't have to write it out. But you do have to write this. So on your pages, it says DNA colon and RNA colon. So here's what I want you to note. So first of all, the sugar is called a deoxyribose sugar, and that's why we call it DNA. For RNA, the sugar is called a ribose sugar, and that's why it's RNA, okay? Now, your bases, again, are adenine, thymine, cytosine, and guanine, right? And here we have adenine, uracil, instead of thymine, cytosine, and guanine. So uracil is in RNA, and thymine is in DNA. Bottom of the page. So again, you already wrote in some stuff here, so I don't know if you need to write all this, but again, for deoxyribonucleic acid, the four that make it up are cytosine, guanine, adenine, thymine. For RNA, cytosine, guanine, adenine, and uracil, right? The one thing I do want you to note here, though, is that DNA is the genetic code for all living things, right? It's double-stranded, the whole deal. RNA is single-stranded, and so what it does is it allows us to copy DNA, but actual, like, inside of you as a human being, your genetic code is actually DNA. And I just wanted to note that. And that is actually it, folks. We have made it through all of the notes that go in our packet, okay, for macromolecules. So that was pages 16, 17, 18, and 19. The last thing you want to do is you want to take this, you want to bring it up to Mr. Gerbe, or bring it up to me, and let me see that you have completed all the notes. Good job.